Thank you very much, Lynn, and thank you to everybody for having me here today. It's a great honor. It's quite intimidating to see the list of other speakers here. I don't really think I belong in that list, but I'm very honored to be here nonetheless. And I've had a wonderful day speaking with a lot of people. Some people I know, I've gotten to know some people too. So I very much appreciate being here, and thanks a lot. I hope I can tell you something useful. Now, one thing I've gotten worried about since I've been here, and I'm going to ad-lib a couple of things quickly, is it's always difficult when I give a talk like this to know exactly how much background in basic behavioral and twin genetics to go over. And the risk, of course, is to talk about a lot of things that you all don't know a lot about. But on the other hand, I want to talk about things that get beyond traditional behavioral and twin genetics. So I'm going to skip pretty quickly over a few things. But before we get to this slide, this slide is about a concept called heritability, which probably to one degree or another you people are familiar with, but I should say just a little bit about it. Heritability is a concept from quantitative genetics. It's a number that varies from 0 to 1 that, well, one can give a whole talk explaining exactly what a heritability coefficient means. It's for those of you with a little behavioral science background, it's a percentage of variance explained, and it tells you how much of the variability in a trait is associated with differences in genotype, simply. And I've made a career, among other things, up to now criticizing the concept of heritability. A lot of behavioral genetics over the years has been about, well, what's the heritability of X, and what's the heritability of Y, and what's the heritability of intelligence and personality and schizophrenia, and kind of assigning all those numbers. And I've spent a lot of theoretical time talking about how that's probably not a very useful thing to do. So I won't read you the whole talk, but the reason heritability is a problem is that it's a variable quantity. The heritability of having 2 arms is 0. And the reason the heritability of having 2 arms is 0 is because we are not genetically variable for having 2 arms. Whatever the genetic background is for having 2 arms, we've all got it, and that's why hopefully all of us or most of us here have 2 arms. Right? But one wouldn't want to conclude from that our genes don't have anything to do with an explanation of why we have 2 arms. Of course they do. It's a very biological construct. But the problem is it doesn't vary. So, well, I'm going to talk a lot about body size. So down the second half, among cloned animals with widely varying diets, body size is perfectly environmental with a heritability of 0, right? Because they're cloned animals and they're all genetically the same. In genetically variable animals raised in identical environments, heritability is 1.0 because their environments are all the same and their genotypes vary. This is no mere statistical fine point. It means that the entire project of assessing how essentially genetic traits are in terms of measured heritability coefficients is a fool's errand. So I stated that quite strongly, but I just want to start my talk off with the idea that it's not true. And it's not true in an important sense that is really the idea that I want to explore in this talk today. And I'm going to talk about this in terms of height and weight. And I actually don't mean height and weight. Height and weight has a nice ring to it, and I might cross over. But anytime when I'm talking about data, I'm actually talking about height and BMI, body mass index. Weight is, of course, very, very closely related to height. So usually when you're talking about height and something, you talk about height and body mass index. And I want to start out by talking about a very simple intuition that we all have about the role of genetics in our height and our weight, our BMI, that is the kind of thing you're not supposed to talk about as a behavioral geneticist, actually, but which I think is an absolutely important intuition that I want to explore in the rest of the talk. Okay, if you think about how you came to be as tall as you are, well, even putting it that way is kind of a strange question because there is no way you came to be as tall as you are. It just happened to you, right? Fortunately, we in the modern world, very few of us grow up under conditions of starvation that might have an effect on our height. So our psychological experience of our height is that it's something that just happens to us, right? It seems very natural for us to say our height is genetic. And from a technical point of view, that's kind of a terrible thing to say. You're not supposed to just say that things are genetic. But of course, from a psychological point of view, that's true. We didn't do anything to become the height we are. It just happened. Right? Our BMI, on the other hand, is a different and more complicated thing. Right? We all recognize that we have a body type. And I think most of us recognize that our body type probably has something to do with our genes. But on the other hand, our body type and our BMI is something that's under our continuing psychological and behavioral control, right? Any adult has spent a certain amount of time trying to create a BMI and trying to become a BMI, to bulk up or slim down. And BMI has that interesting quality that I think is at the heart of behavioral genetics, that it's at the intersection of a genetic hand that we're dealt and then a behavioral process that goes on from there for the rest of our lives, where we try and play that hand and in a behavioral way do something about our BMI. And the idea I want to talk about is that distinction between things that just bloom in us from a biological point of view and other things that are always filtered through our own psychological and behavioral control are what behavioral genetics is really about. Okay? Because if that's not the case in the modern age, there's really no need for there to be a behavioral genetics. The basic methods of genetics apply to behavior just as much as they apply to anything else. Twin and adoption studies that I'm going to talk about, you can do for medical conditions, you can do it for diabetes, you can do it for height. Right? There's nothing behavioral about them. And as we've developed more modern DNA-based methods for behavioral genetics, those apply from a scientific point of view to behavior in exactly the same way they apply to biology or any kind of physical characteristic. What makes something behavior and what makes it a topic for behavioral genetics is that it's experienced through our own psychological experience of it, which is why I call it intentional genetics. Behavior genetics is genetics intersecting with our human intentions. So what does intentional genetics look like? So what happens to the genetics of a trait when it is filtered through our own psychological experience? One thing is that intentional behavior is often skewed. That's a simple plot of height and BMI in one of the samples I'm going to be talking a lot about, the University of Washington Twin Register. And height is normally, very nicely normally distributed. BMI in a population sample is very strongly positively skewed. And if you're a statistician, it's very common to think, well, we should just take the log of that or something and make the skew go away. But I actually think the skew is important. I mean, if you think about why is it that in the modern world a lot of people become heavy but very few people become skinny, it's an interesting question, and we'll return to it. Well, it's also the case, and this is something that I've spent most of my career talking about, that intentional behavior of all kinds is heritable. And I think one of the basic incorrect, as it turns out, intuitions that people bring to behavioral genetics is that maybe very biological things like height are highly heritable, and you can really see the action of our genes in biological things like height. But for more psychological things, of which, of course, weight and BMI is just the beginning, and moving on from there to personality and intelligence and all those behavioral things, maybe we don't see the effects of genes in that. And I've spent, oh, well, that's funny. That's not where I wanted that slide. That's just let's just skip it. Never mind. I've spent a career making the point that, in fact, everything is heritable. Everything about human beings all the time. If it varies, if it's not like being two arms, if we're not all the same, then it's heritable. Right? And the history of behavioral genetics has been about discovering that many of the obvious psychological things that you might want to look at when you started wondering if behavior is heritable, like all the major psychological constructs, intelligence, personality, schizophrenia, depression, bipolar disease, proceeded down to sexual preference, which 10 or 15 years ago turned out to be heritable. One after another, and in the early parts of my career, twin and adoption studies showed one behavioral trait after another was heritable, and people were amazed that, wow, the world is turning out to be a much more genetic place than anybody ever imagined. But a funny thing happened. It turns out that everything is heritable. Political attitudes are heritable, liberal versus conservative. How much television you watch is heritable. The kind of food you like to eat is heritable. How often you vote is heritable. Your economic preferences are heritable. All human behavior is heritable. So whatever it is that genetic influence on behavior is about, it's not about dividing the world up into some things that are genetically based and other things aren't, because all differences among us are, at least in part, related to our differences in our genes. An interesting secondary fact about intentional behavior is that, by and large, it doesn't fit twin models. And actually now, I'm going to go back up to that slide. This is not a talk about the classical twin design, and I'm personally bored with the classical twin design. But we should establish a little common language that'll help me talk to you. A lot of behavioral genetics, as a branch of quantitative genetics, is based on the analysis of different levels of biological relatedness in families. And in particular, it's related to differences in similarity between identical and fraternal twins. And I'm going to do this very quickly. I think you all are familiar with the idea that there are two kinds of twins in the world, identical and fraternal. Identical twins share 100% of their linear sequence of DNA. Fraternal twins are ordinary siblings who happen to be born at the same time and therefore share half of their DNA sequence, like any other pair of siblings. Doing twin analysis of identical and fraternal twin pairs allows us to decompose the variability in a behavior into three parts that are the three variance components of the classical twin design. The first of those, A, which stands for additive, is the genetic part. And it's the additive effect of multiple genes that make people in the same family who share those genes alike. And identical twins have twice as much of that as fraternal twins do. The second part of it, C, which stands for common, refers to the common family environment. Right? And the common family environment is the aspects of the, sometimes called the shared environment, the aspects of the environment that are shared by siblings raised together in the same family and are environmental effects that make siblings raised together more alike. The third part, E, which actually stands for environment, is the so-called unique or non-shared environment. And those are the environmental effects that make siblings raised together in the same family different from each other. So a common way to talk about the results of basic classical behavioral genetic studies is in terms of A, C, and E. The percentage of variability that's attributable to additive genetics on the one hand, and then the common and unique family environments. Well, a conclusion of models like that, since identical twins are twice as similar as fraternal twins, if a trait is completely determined by genetics, well then identical twins ought to be twice as similar as fraternal twins. But an interesting thing happens for intentional behavior, which is that generally it is commonly true that identical twins are more than twice as similar as fraternal twins, which is a problem for the classical twin model. This is just a very simple illustration of something that very commonly happens. This is from a large American, I'm not going to talk much about my data sources because I have other points to make. This is from a large contemporary American sample that includes twins and siblings called the Adolescent Health and Development Project. We refer to it as AdHealth. So these are the twin correlations for BMI and height for MZ and DZ twins. And if you start over on the right for height, you see what you would expect, which is that MZ twins are a lot more similar than DZ twins for height, but they're a little bit less than twice as similar. And that's an indication that if you double 0.53, you get 1.06, and the difference between that and 0.84 is an indication of a shared family environmental effect on height, which I'm not sure exactly what that means. But in any event, it fits the twin model. It's mostly genetic, and there's some environmental effects left over. BMI does something it's not supposed to do. Identical twins are as correlated for BMI as they are for height. But fraternal twins are correlated much, much lower than fraternal twins are for height. And in fact, they're correlated less than half of the identical twin correlation, which is a violation of the twin model, and it's a problem. And I'm going to spend a lot of time today talking about what happens to fraternal twin correlations for intentional behavior. It gets driven down. A consequence of that is that it's very, very difficult to find the effects of the shared environment in twin studies. One of the great crisis that twin studies have presented to psychology has not been so much that they've shown that a lot of behavior is heritable. People have gotten pretty used to that idea. But that in general, twin studies suggest that your family environment doesn't have anything to do with behavior. I mean, I don't think that's true, but when the MZ twin correlation is more than twice the DZ twin correlation, that's what it's supposed to mean. So when that happened for intelligence, for example, and I'm going to be talking about intelligence later, when it turned out that identical twins were often more than twice as similar as fraternal twins for intelligence, it suggested that how your parents raised you had nothing to do whatsoever with how your intelligence turned out. And that was a troubling and difficult finding. I've referred to that in the past as the second law of behavior genetics. And the question I want to ask is, why is it that identical twins are often more than twice as similar as fraternal twins? So, because twins are not perfectly correlated, not everything is explained by genetics, and because families within, for the time being, in the context of traditional twin studies, don't explain anything else, that means that the bulk of what's left over, what environmental effects there are on intentional behavior, are of the non-shared variety. There was a famous paper by Robert Plomin and Denise Daniels 20 years ago, sort of calling attention to this fact, calling attention to the idea that whatever it was that whatever the means were by which the environment was affecting behavior, it wasn't doing it via families, it was doing it independently to children raised in the same family. So whatever the environment was doing, it was making kids in the same family different. So they said one implication of our conclusion concerning the importance of non-shared environment is that environmental factors shared by both children in a family are unlikely to be important sources of environmental influence. And if you think about it, those environmental sources shared by both children in the family are most of the top things you think of when you think about the environment, like poverty, like socioeconomic status, like your parents getting divorced. All those things are things that are shared by children in the family. So they said, well, what is it? You know, what are these things that are not shared by children raised together in the same family? And they outlined a three-step research program to begin with quantifying the overall effect of the within family, which is another word for non-shared environment, because it operates within families, identify the specific environmental variables within families. What is it about how parents treat their kids differently, for example, that winds up making them different? And then finally, finding the causal associations between that within family environment and behavior. But it turns out, this led to their credit, to Plomin and Daniels' credit, they published this paper in about 1990, and it led to 20 years of empirical research on this topic that had a very interesting and sort of troubling outcome, which is that when you actually study actual measured behaviors within families to try and figure out how children's environments differ within families and how that causes differences in behavior down the line, there's nothing there. Nobody could find anything. At the end of all this, finally coming to something that I did, my student Mary Waldron and I published a comprehensive review of this whole literature. This is of studies, for example, of how differential environmental factors produce differential behavior in children. Right? We did a lit search. I don't need to go into those details. So the first thing we did was simply to quantify the variance of the magnitude of the variance components. And this is what I've already talked about, that a substantial portion on average across a very wide range of behaviors An average of 40% of that was attributable to the additive effect of genes. Almost none of it was attributable to the shared effect of families, C. And the rest of it was attributable to the non-shared environment. That's what Plomin and Daniels had already observed. And those 3 bars add up to 100% of the variability in behavior. But when we tried to divide that E bar up into specific behaviors, how mom treats sibling 1 as opposed to how mom treats sibling 2, a funny thing happened. It dried up and blew away. Of that 40% of the variability that's attributable in an overall twin sense to the non-shared environment, the median effect size for how much was attributable to a measured environmental effect was less than 2%. So something is making kids in the same family different, but when we try and specify what those particular behaviors are, we can't find anything. I have a whole other talk that I'm not giving today about parallels between that problem. You could call that problem the missing environment problem, right? You might call it the missing environment problem because we know there's this big thing out there called the environment that makes kids in the same family different from each other. But when you try and figure out exactly what it is about the environment, it's missing, it's not there. And a very interesting thing has happened in recent years that I'm going to have to skip over way too quick. The same thing has happened in the genotype. And in fact, I borrow the missing environment problem from the contemporary term that's quite familiar in the literature, which is the missing heritability problem. And an interesting thing has happened in the last 10 years. When I started in the field, behavioral genetics and this kind of genetics in general was based on twin and adoption studies, say. I haven't talked about adoption studies, twin and family studies for a reason, which is that we couldn't get to the DNA. Right? I mean, we knew people had DNA in 1985 when I got started, but we couldn't really, in a useful way, study human DNA. And now, since the completion of the Human Genome Project, we can. Right? We can, in a variety of ways that I'm not going to get into, we can actually collect human DNA and face the exact same problem that they faced in the non-shared environmental literature. That is to say, well, we know, say, 40% of the variance in behavior is accounted for by this big unspecified thing called genotype. Can we now break down that effect into the effect of individual genes and find out what are the genes for schizophrenia? What are the genes for intelligence? What are the genes for personality? And if you follow this literature, you know that that attempt has been a surprising failure. We have not, by and large, been able to identify any genes of large effect for most interesting psychological characteristics. I thought since I'm talking about BMI, I would just give there's missing heritability for everything of interest. But missing heritability for BMI is a big problem. Remember, identical twins from my first slide are correlated almost 0.9 out of 1 for their body weight, their body mass. But if you search through the genome, and I'm not going to go into the technology, it's something called GWAS, a genome-wide association scan, which searches through DNA for any little snippet of DNA that might be associated with body mass index, you cannot find out of that 90% that's accounted for by genotype, you cannot find a piece of DNA that accounts for more than maybe 0.5%. And if you add together all the little snips of DNA you can find, you can account for maybe 2 or 3%. And the rest of it's missing. Right? We don't know where it went. Okay, some more things about the heritability of intentional behavior. A very common observation in behavioral genetics is that the heritability of intentional behavior, the difference between MZ and DZ twins, or very roughly the importance of genes for differences in a behavior, increases as people get older. That is a graph of the genetic variance of BMI in the University of Washington Twin Register, which goes up by about 20 or 25% between the ages of 20 and 80. So whatever else is going on, as people age, the proportion of the variability in their BMI goes up over time. In addition, the heritability of intentional behavior varies at the extremes of behavior. So one thing I've been very empirically investigating very intensely lately is how the heritability of BMI varies by level of BMI. So this plot is of the correlation between MZ and DZ twins. And I'm sorry, I'm missing a label there. The top line is the correlation between pairs of MZ twins. And the bottom line is the correlation between DZ twins. And that correlation is varying as a function of the mean BMI of the pair. And one thing I should say to help you, for those of you who aren't familiar with BMI, I had that plot of BMI. The average BMI is about 24 in women and 25 in men. There are very strict government standards these days. Over 25 is actually considered overweight. My BMI, for reference, is just about exactly 25. Over 30 is considered obese, and over 35 is considered very obese. And what you see, so the fact that identical twins for most of that graph are more similar than fraternal twins is an index of the heritability of BMI. Identical twins are more similar. But as BMI increases, as the average BMI increases, those correlations go down, especially in the DZ twins. Right? And by the time you get to the right, in very heavy people, the twins are not correlated at all. The correlation is 0. So there's absolutely no relationship between an identical twin with a BMI over 35 and the probability that his or her co-twin is going to have a BMI over 35. Another characteristic of intentional behavior is that it's attenuated in unfavorable environments. This is some work, again, in the University of Washington Twin Register that I've just finished. The black region there is the genetic variance in BMI. And here we've plotted it against the number of minutes of moderate to vigorous physical activity that people report. And moderate to vigorous physical activity, surprisingly, has a very weak effect on the mean of BMI. People can exercise quite a bit without having a significant effect on their BMI, but it has a powerful effect on the genetic variance in their BMI. For people who exercise a lot, express much less of their genetic variability than people who don't exercise. So, a quick summary before I move on. Heritability in itself doesn't matter, but some forms of behavior show a characteristic pattern. And maybe I won't use up my time really just listing everything that I've said. And I now want to explore what could cause that. And I'm going to suggest that this is going to be the most technical part. So there's a single parameter in developmental behavior genetics that can account for all those characteristics of intentional genetics. And that's what I call P to E effects. P stands for a genetics word called phenotype. And all phenotype means is an observable characteristic of an organism, as opposed to their genes or environment. So BMI is a phenotype, and intelligence is a phenotype. So the idea of P to E effects is that changes in our phenotype have an influence on the environments to which we're subsequently exposed. So some of this is for the behavior genetics crowd. This is a characteristic that's often thought of as G to E effects, genotype by environment interactions. But our genotypes can't really affect our environments. If the way a kid behaves affects how mom treats him, the genes aren't reaching out and magically affecting mom. It's the kid's behavior, his phenotype, that's affecting mom. I'll leave most of that. Here's an example, skipping ahead, actually, into the intelligence section of my talk. I should make a BMI one of these, for how P to E effects work, particularly within families. So I'm talking about within family P to E effects. And we're going to imagine two siblings. my student Chris Beam made this slide and always refers to them as the bright twin and the dim twin, but that's not a very nice way to put it. So we have sibling 1 and sibling 2. And I want you to imagine that for reasons that might be completely random, there is a small difference in ability at an early age between sibling 1 and sibling 2. Maybe there's some minor genetic difference between them. Maybe one of them had a serious illness when they were young. Who knows? It could be anything. But sibling 1 and sibling 2, they have a small difference. And sibling 1, the smarter one, because he seems a little smarter, gets placed with a good math teacher. Whereas sibling 2 gets placed in with the not-so-good math teacher when they notice that he doesn't seem to be performing as well. And then for the bright twin, because he's in the good class, he becomes even better at math, which makes math more fun, which gets him higher test scores, and that kid eventually goes to MIT. Whereas the dim twin takes the opposite path. From that lousy math teacher, he's worse at math, he enjoys math less, gets lower test scores, and winds up in bartending school. Right? And the point of this story, I mean, it's obviously sort of a silly story, but the point of this story is that feedback loops between our behavioral characteristics and our environment can take what are initially very, very small differences and over time expand them. These are the kind of models that Chris fits for me. And a lot of these are pictures of actually simulations rather than data analyses. And I'm going to make a long story short here, is that Chris and I have shown that if you introduce P to E into developmental family studies, if you allow your behavior at time 1 to influence your environment at time 2, you can reproduce all of those odd characteristics of intentional genetics that I listed for BMI. And what this is showing under 2 different conditions of P to E what happens to identical and fraternal twins over time. Identical twins on the top are relatively unaffected by it because they're so similar to start with, there aren't very large differences to get that P to E engine going. But fraternal twins, who are more different to start with, as time goes by, they become less and less and less similar. Right? And as the difference between the MZ lines and the DZ lines gets greater and greater, the heritability is going to be going up and up and up, which is, of course, exactly what you observe. So models that have P to E in them, and again, I think I'll make this short, reproduce all of the characteristics of intentional genetics. In particular, I think that one down there is the important one. Environmental effects are observable in terms of increases in within-pair variance. That environmental effects set off developmental cascades that push DZ twins apart at faster rates than MZ twins. It also explains, by the way, well, I'll wait, I'll explain that in a minute. Complex reciprocal developmental processes blur the effects of individual genes and environments. And again, this is a little technical, and I'm going to do it fairly fast so I have time to finish. It's something I wanted, this is work I did a long time ago and don't often get a chance to cite that I think is kind of fun. It's a different kind of simulation. I simulated little genetic and environmental systems. These little organisms that I made have 10 genes that you can see floating around in the space there. They have one phenotype, the P in the middle of the space, and a changeable and movable environment, that E that up there is in the lower left of the box. And this system is designed so that all the genes and the environment have an effect on the phenotype. They make the phenotype move. And the phenotype, going down to the lower left, also has an effect on the environment, which in turn has an effect on the phenotype. So there are P to E effects built in here. And you set this up and let it run. And the little phenotype has a life. It zooms around the space, and it eventually settles somewhere. And you can then do studies to find out where this thing ended up depends on where its genes are and where its environment is. And what's interesting about it is that you basically reproduce the effects of twin studies when you do this. There is a nice linear relationship between the average genotype and where the thing ends up on the top. That's a nice effect of genes. You get no effect of family environment whatsoever, even though I built family environment into this thing. I mean, it's there, but inside the system, because the environment is free to move and change as time goes on because of P to E, the effect of environment is impossible to see. Well, and this is just show this. It looks exactly like the data for BMI, that identical twins wind up. I make twins in this system. The little spaces can have the exact same genes, or they can have half their genes, share half their genes. And when you do twin studies with it, the identical twins come out more than twice as similar as the fraternal twins. And the effects of the individual genes are lost. The heritability is all missing. If you say, what's the effect of having a gene right in this particular place, you can't find it. So anyway, in conclusion of this whole section of the talk, the question isn't, is behavior heritable? Because all behavior is heritable. The question is, how do the genetics of behavior interact with human intentionality, which is what P to E is. Okay, let's talk about something a little bit closer to the bone than for behavioral geneticists than BMI, and that's intelligence, where I've actually spent most of my career. I've never, until very recently, been a BMI researcher. Intelligence follows all of the patterns of intentional genetics. The stability of intelligence increases over time. This is just showing us where the data came from. This first analysis comes from something called the Netherlands Twin Register. And I'm going to show you a very particular parameter here that I think is very interesting. And what these are, and I'll say it first and then explain what it is, longitudinal within pair correlations between identical twins. And the idea is this. These are IQ scores. And these are young twins growing up. And within identical twins, the within pair score is who's smarter, twin 1 or twin 2. So it's as though you subtract twin 2 from twin 1. Okay? And the question we're asking is, within pairs of identical twins, if twin 1 is 3 points smarter at time 1, how does that predict which twin is going to be smarter at time 2? Okay? And the answer is it doesn't. Identical twins switch back and forth as to which twin is smarter almost at random in childhood. And that's those close to zero correlations you see there. Almost at random. That parameter is, in fact, it's often reported as though we're zero. It's not zero. It's just a very small positive number. If we do the same thing, though, in adult twins, these are from the Swedish adoption and twin study. These are people who are between ages 40 and 90. And we do the exact same thing. That is, look at differences at time 1 and see how they're related to differences at time 2. That same parameter that was basically indistinguishable from 0 in little kids has become indistinguishable from 1.0 in adults. Right? So if you're smarter than your co-twin at age 40, you're still going to be smarter than your co-twin at age 80. Right? So intentional behavior stabilizes over time. The heritability of intelligence, like the heritability of BMI, except much more familiarly so, increases with age. This is not my slide. This is from a famous behavior genetics researcher named Tom Bouchard. And that's just, the graph runs from 5 years on the left to 50-year-old adults on the right. And the heritability of intelligence increases from about 0.2 in little children up to getting close to 1 in adults. Just my analysis of the same phenomenon in the Adolescent Health and Development Project, where there's not quite as wide an age range. These are kids growing up from age 12 to 12 on the left all the way over to age 24 at the right at 2 different waves. The black area is the genetic variance in a very simple intelligence score in Ad Health, and it, once again, increases with age. And what I want to think about quickly is why this happens. Behavioral geneticists think about this in simple terms as just heritability going up with age. But if heritability increases with age, that's because either identical twins are getting more similar or fraternal twins are getting less similar or both. Well, I feel like I'm missing a slide there. But the answer is fraternal twins get less similar over time. Identical twins start out similar and stay similar, while fraternal twins drift apart from each other over time, which is exactly what the simulations would predict. Like other intentional behaviors, most of the heritability of intelligence is missing. And this is a much more famous phenomenon than the missing heritability of BMI, because people were and are you can imagine that finding genes or DNA that are related to human intelligence would be a big deal. I mean, not necessarily a good thing, but it'd be a very complicated thing. And people have looked very, very, very hard, and they found nothing. This is a report that was actually trumpeted as a positive result, which gives you an idea of how this field goes, of finding a SNP, which I won't bother explaining, something related to a gene for intelligence. And each of the genes that the SNPs, the single nucleotide polymorphisms that they found had, for those of you who know a little statistics, an R-squared value of 0.0006. So just think in your own research of trying to get something into PNAS with an effect size of 0.0006. You add them all together, they accounted for 0.0002 of the variance. And that's just to say it's missing. It's not there. The Louisville twin study. I know I'm jumping around a lot with data sets. I study this in a lot of the Louisville twin study, we recently got a grant. The Louisville twin study is one of the most important twin data sets that was ever collected back in the 80s and 90s. It sort of came to an unfortunate end. The lead investigator died, and it lost its funding in 2000. We now have funding to go out to Louisville and recover all the old data. It's very exciting. It's the most intensely longitudinal twin data set of intelligence and a lot of other things that has ever been collected. They have a fairly large number of twins who were repeatedly tested between 3 months of age and 15 years old, up to 18 times. Very, very intense. Actually, they have a lot of good growth data, too. What happens this is a plot of the MZ on the top line, DZ on the bottom line, twin correlations over age, and they look exactly like the simulations. As the Louisville twin studies grow up, the DZ twins grow apart and the MZ twins stay about the same amount similar, so the heritability of intelligence increases between 3 months and 15 years. And in fact, the increase in the heritability of intelligence over time is sometimes known as the Wilson effect, after Ronald Wilson, who started the Louisville twin study, because that was the first place it was observed. The heritability of intelligence is attenuated in unfavorable environments. As Wendy pointed out, this is my most cited paper from 2003, showing that we found a very unusual data set that had a very large number of very poor American twins in it. So that's socioeconomic status on the x-axis of those plots, A, C, and E, and the proportion of variance attributable to A, C, and E on the y-axis. And as you move on, if we look on the A box over on the left, as we move from the poorest families on the left, where almost none of the variability was accounted for by genes, to the middle class and up families on the right, almost all of it was. So something was going on in those poor families that prevented the expression of the genetic variability in intelligence. That finding's been replicated a lot since then. I won't bore you with it. It happens in the Adolescent Health and Development Project, where they have more than just twins. They have identical twins on the top line, fraternal twins and siblings, ordinary siblings on the middle line, and then things like half-siblings and cousins on the lowest line. And the x-axis is parental education, which is our proxy here for socioeconomic status. And what you see as you move from the poorest educated families to the best off families, the identical twin correlation, once again, is not particularly effective. What happens is that the DZ twins, and even more so the less related people, the half-siblings and the cousins, become more and more different in the good environments, the facilitative environments. And because the identical twin and the fraternal and less related lines diverge, the heritability goes up. Finally, I wanted to get to at least some data that I haven't presented elsewhere before. A problem with these poverty by heritability studies that I've been showing you is that obviously in human beings, well, I'll close with this idea in a minute, but we don't really know that socioeconomic status is just, quote, an environment. There are we cannot randomly assign children to socioeconomic status. People come to their socioeconomic status by a whole lot of routes. Some of them are even related to genetics in a very complicated way. So it would be nice if when we study mice, we can randomly assign mice to environments. And we normally think of it as being impossible to randomly assign humans, and especially human twins, to random environments. But it turns out there was a study that did that. Something called the Infant Health and Development Program, called the IHDP. I don't know if anybody's familiar with it. The IHDP was something called a randomized clinical trial that was conducted in the late 90s in an era when what people were interested in was the effects of randomized change in environment on the average level of IQ. So the question is, if you take children and at random assign them to a special Head Start program, what kind of effect does that have on their IQ? And there were some very successful studies. This was designed as a follow-up. It was of low birth weight infants who were assigned at random to either a very intensive special educational program for both the parents and the children for 377 families, or 608 control families who just had regular medical care pediatric follow-up. Now, again, I don't know whether any of you are IHDP people. It was a very well-known and in many ways very successful study and that produced a lot of research. It did not produce a very big effect on IQ. It was a bit of a failure in that regard. The effect of the randomized special intervention on IQ was on the order of a couple of IQ points on a good day. And under many circumstances, you couldn't see it at all. It's kind of faded from view since that time. But an interesting thing, it turns out, and I credit my colleague Clancy Blair with noticing this, that for pretty much random reasons, the IHDP included 95 twin pairs of known zygosity. And these twin pairs were always assigned as pairs to the same condition. There were MZ and DZ twin pairs. 39 of them, is a very small N I'm reporting here, 39 of them were randomly assigned to the intervention condition, and 56 of them were assigned to the control condition. A lot of background variables that I won't bother you with, and we don't need to look at those numbers. Here's what this slide is telling you. The IHDP didn't have any big effect on the mean IQ in its full sample. It had absolutely no effect on the mean IQ of the twins. None. So why am I telling you about this? It had a very interesting effect. In these 80 or 90 twin pairs, well, we can ask, what happened to the heritability of intelligence, the difference in similarity between the MZ and the DZ twins in the enhanced treatment group and the unenhanced control group? And lo and behold, that's exactly where the effect turns up. Is that red showing up? Yeah. So I should say one more thing to say about the IHDP. The intervention lasted until the kids were 3 years old and then ended. Okay. They were then followed up until they were 8 years old. And from right here down are the 8-year-old tests. And the heritability on every single test given at 8 years old was higher in the children randomly assigned to the enhanced environment than it was in the children in their unenhanced environment. And the reason that happened is exactly because in the treatment group, the DZ twins became more different. So in the facilitative environment, the DZ twins drifted apart, exactly like they do in the SES. But these environments, of course, were not selected. They were randomly assigned to these environments. For those of you out there who report data, this is we've actually had trouble getting this published because it's a very small sample of twins. These results are exactly what my theory of this would predict, but they're none of them statistically significant. I still think it's true. So, a summary. Outside of the lab, human beings select their own environments, and as a result of that, genetically related people become more different over time at a rate inversely proportional to their degree of relatedness. Fraternal twins become different more rapidly than identical twins do. Identical twins are special because they're so similar that they don't have that differentiation mechanism that gets the process of differentiation started. I think I'll leave that. I know I have quite a few slides left, so I'm going to keep moving. Actually, I'm in pretty good shape. I want to end with some very general thoughts about why all this matters. There was a very famous exchange about this, what turns out to be this topic a long time ago in 1967. A medical geneticist named Roberts said the following: the genotype may influence the phenotype either by means of biochemical or other processes labeled for convenience as development or by means of influencing the animal's choice of environment. So the genes can affect behavior directly, or they can affect behavior by getting mom to treat us differently. Right? But this second pathway, just as much as the first, is a genetic one. Formally, it matters not one whit whether the effects of the genes are mediated through the external environment or directly through, say, the ribosomes. Okay? This is a very biogenetic view of what it means for genes to have their effect through the environment. To which my mentor, John Lowland, replied, first, although formally it may not matter one whit in which way the effects of the genes are mediated, in practice it often matters quite a few whits, especially if one should happen to be interested in intervening in the process. Changing behavior by changing parental attitude is a decidedly different proposition than tinkering with the ribosomes, even though a similar behavioral change might conceivably be brought about by either means. So it matters. It matters whether our behavior is intentional and whether the eventual influence of our genes is filtered through our intentional behavior or not. In addition, most of the behaviors that we're actually interested in, what we're seeking in them is stability, not zero. Right? So if we're trying to control BMI, if we're interested in weight reduction, we're not trying to make human beings as skinny as humanly possible. We're trying to get people to establish a stable weight that they can maintain over time. That's true for many different aspects of intentional behavior. Alcohol use, externalizing, depression. We're not trying to make everybody as cheerful as humanly possible. We're trying to keep people stable in their affect. Right? Intentional behavior seeks a stable balance of phenotype to environment processes. In addition, many positive and negative interventions are like the IHDP. They show their greatest effects much later and in disparate domains. One of the characteristic findings in the randomized intervention literature, in something called the Chicago Preschool Study, where they randomly assigned young children in Chicago to enhanced preschools or to control conditions, it once again had no effect on the mean at the time. It was a big disappointment. Except it turned out 20 years later that the children who'd been in the enhanced condition were less likely to go to jail, were earning more money, that their P to E processes down the road had led them in positive directions. Finally, I think it matters for one very important reason. What characterizes me as a scientist, I always think, is that I'm not an experimentalist. And those of you who are in the sciences know this distinction. Science is a whole lot easier when you have, this sounds obvious, when you have experimental control over your phenomenon. Right? And those of us who work in complex behavior in human beings don't have that. Right? If you're interested in why some kids become juvenile delinquents, you cannot assign some children at random to bad environments and see what happens to them. And behavioral science would be a whole lot easier if we could do that. And sometimes, take it from me, it seems impossible to do science under those conditions. I have moments of very profound frustration when it seems to me quite impossible to understand why complex human behavior comes out the way it was when I don't have experimental control over what I'm trying to study. And I think the fundamental reason for that is that we can't conduct randomized experiments on humans. The fundamental working method in science is unavailable to us. And I want to close by asking just a sort of an odd question. Why can't we do that? Why can't we? Why can't we randomly assign children to alcoholic and non-alcoholic parents and see what happens? It works great in mice. Why not? Well, easy to say it's ethically impossible, but what does that mean? Well, the reason we can't is because there's an aspect of human behavior, and I say this as a scientist, that we regard as sacred. Right? That people's ability to choose their own environments, their P to E, if you want to call it that, is the characteristic of human behavior that we hold most closely. Right? So the very thing that prevents us from doing science is the aspect of human beings that we feel the most strongly about. And because of that, some aspects of human experience remain seemingly forever beyond simple mechanistic scientific explanation. Right? So to me, all of these things form a circle that describe to me what behavioral genetics is about. Behavioral genetics is about the attempt to do science on the kinds of behaviors on which we're in some sense not really allowed to do science. And that's both the great frustration of being a behavioral science, a human behavioral scientist for me, but it's also the great attraction. I'll leave you with that thought with thanks for all my many collaborators. Thank you very much. And Ken, I added that for you. Yes, questions?
I wanted to come back to the issue of positive feedback
Yeah.
over time. It seems to me that either across domains, across time, or across, let's say, social groups, there would be differences in beliefs, sort of folk notions of what causes what in terms of, let's say, nature and nurture. So I could imagine some domains in which you actually get negative feedback. So if the belief is that nurture matters more than nature, you get interventions that would point against open spreads. Whereas if the belief were the differences are due to some natural cause, then the differences that open up just reify this belief, and so they would sort of continue. So I'm wondering if in some domains you might get less of this opening up versus others, or across places, which could be due to sort of negative feedbacks as opposed to positive feedbacks.
Yes, no, it makes sense, and I agree that it could happen. And one thing I will admit to sort of glossing over here is that really the message here is that once you allow those kind of feedback loops in your model, the most profound effect it has is make everything less predictable. And it doesn't take a very large change in a small parameter to change things from spreading out to closing in on each other. And I mean, it's a danger in these models because it means I can kind of sometimes post hoc explain almost whatever happens. But on the other hand, I don't think I'm suggesting that it's the phenotype that makes things happen. So if I notice that one of my children, and those of you who are parents, I mean, I think we parents all have this experience. One of my children is a much better athlete than any of the other three, and any of the other two. And I invested way more time and effort and coaching in that kid's athletic performance than I did in any of the others. And that has magnified, right? This kid, maybe he had a little bit of a genetic advantage. Again, who knows? I don't think it matters. It was from the day I went out in the yard to play ball with him, it was more satisfying for me, it was more satisfying for him, so we did it again. And then it was more rewarding to spend the money on the special coach, et cetera, et cetera, et cetera. So my point is that I'm not sure that that activity on my part really depended on whether I believed that the original difference was genetic or environmental or nature or nurture. It was a response to the behavior itself, that it was just satisfying to invest resources in that kid's athletics, where it was satisfying to invest resources in another kid's social life or whatever. So I agree with you, but I don't think all of this depends on our belief that something is nature as opposed to nurture, though just one final thought, there's an interesting literature on that, that if you give people a lecture about how weight is fundamentally under genetic control and leave them in a room with cupcakes in it, they eat a lot more cupcakes than if you tell them weight is largely up to us. So those things do have an effect. Yeah?
Real quick, but what I meant was, let's suppose you observe one of your kids slightly larger than the other one because maybe they're eating more, and you were not a non-human animal, you might, let's say, preferentially feed the other one more. You wouldn't necessarily keep feeding. Sometimes you would, sometimes you wouldn't, but it depends. That's all I meant by that, where if you sort of had negative interventions because of what your goals were, then you would sort of over time squash these things. But I see your point.
I agree. Yes. Other questions, please. Yeah.
I'm not quite sure this all integrates into the model, but the business about, say, obesity or diabetes, immune disease, you're only finding 1% heredity, okay, genetic causes, you're doing the SNPs.
Diabetes is going a little bit better than obesity, yeah. Yeah,
Cancer
no,
or whatever, okay. They explain that away because they've got a whole lot of money invested into over 30 years of genetics. It's a big deal that we should know the sequences, that it isn't just individual SNPs, it's multi-genetic.
Right.
Multiple genes that are involved in that could explain why it could be genetic to a much higher level. Just they're not analyzing for multiple genes because of the power of their test. So how does that fit into your model about balancing heredity and environment? I couldn't follow that.
Well, it's important to recognize that my conclusion from missing heritability is not that the twin studies were wrong all along and these things really don't have genetic variance. They really do have genetic variance in them. It's that the genetic variance in intentional behavior is filtered through so much psychology that the individual effects of any particular gene are lost in the system. So twins do something very unique. Our behavior would be very genetically predictable if we all had an identical twin, because identical twins add up the effect of all those hundreds of thousands of genes that contribute to complex behavior and combine them in a useful way that show you, I mean, obviously it doesn't work this way, but if we all had an identical twin who could lead a life before we did, and we could observe how extroverted that identical twin was, well, then we could add up all the effects of those genes and make a prediction about how extroverted we would be. But since we don't have that, looking at the genes one way at a time, one gene at a time, just turns out not to work. That the genetic, and I think it's because the genetic pathway between the genes and our behavior is filtered through our psychological intentions, just like those little squiggly diagrams of mine were the effects of those individual genes in the space had to be filtered through the little squiggly lives of all the points as they moved around in there. So those genes more than influenced the course of those paths, they determined it. I built the program. But still, at the end of the day, if your job was figure out exactly how each gene influenced where that little point came out, you couldn't do it. So it's a level of analysis problem. You can observe these genetics at a complex behavioral level of analysis in twins, but you can't observe it at the individual genomic level of analysis. Yeah?
This is perhaps related. Everything you've said makes sense to me.
Well, that's
in
good.
terms of comparing the monozygotic to the dizygotic twins, I just wondered that there seems to be this assumption, or maybe simplifying assumption, that the genes act independently, whereas if you consider them as an interactive kind of recipe, you could imagine that you'd expect monozygotic twins to be more than twice as similar.
Yes.
Because, for example, maybe some people are interested in the same things they're good at, and maybe other people are interested in things that are different than they're good at. So, why would you expect it to be half as similar?
Well, that's really good point. And in fact, the classical quantitative genetic explanation of why identical twins are more than twice as similar as fraternal twins is something called epistasis, which is gene-gene interactions. And so there's no way to really do this in humans, but in fruit flies, you can show that a certain veining pattern on the wings is related to having all 5 of 5 different genes. And, well, identical twins are always going to share those 5, but in fraternal twins, they're almost always going to be broken up, so you see the same pattern of veining in identical cloned fruit flies, but not in... Yes, you're right. It's an alternative explanation. It doesn't explain the rest of the observations. It doesn't explain why fraternal twins get more different over time and identical twins don't. It doesn't explain why fraternal twins become more different in good environments and identical twins don't. It just hasn't proved to be a very useful concept in human genetics exactly because you can't find a behavioral human characteristic that has that under control of 5 genes quality like you can in the veining of a Drosophila. So it hasn't proved to be especially useful in explaining human behavior. Yep.
Yeah. So we talked about this a little earlier, but I wonder what your thoughts are. One possible implication of what you're saying is that we're just not good at phenotyping too, because there is a filter there. So it's kind of on the other end. Maybe that's why we're not as successful, perhaps, in GWAS studies and so forth, because we have a very vague kind of phenotype that we're trying to tie to this stuff. I wonder what your thoughts are about that.
Well, yeah, I mean, it's something that people have said for a long time, explaining why schizophrenia genetics isn't going better or something. And it's, I think, hard to find a good example of, well, once we figured out this good way to measure X, the genetics all snapped into place. I can't think of one. And in fact, intelligence, as psychological constructs go, is really well measured. I mean, of course, there are a lot of questions about exactly what intelligence is, but from a strictly measurement point of view, we're very, very good at measuring IQ. We're probably better at it than any other complex behavioral trait. And it's still been a mysterious failure to find anything that, and people have, I mean, those of you in the field know, people have looked incredibly hard for IQ genes. It's a Nobel Prize if somebody finds it. I mean, I think it would be actually a terrible societal problem if that ever happened, but I'm sort of happy it hasn't. But it would be an extraordinary finding. People have run hundreds of thousands of people comparing their IQs and their genes, and there's nothing there.
Can you explain how people can reconcile that paper you did on not being able to find any non-shared environmental factors that accounted for any of the variants? Like, how does that fit into?
Well, I just think it's the same phenomenon. You know, if, so what Wendy is asking is, I think, correct me if I'm wrong, I argue that there are two things in parallel. That we know that a big chunk of the variance in children is attributable to this thing called non-shared environment, environmental effects that make kids different. But when we look for the particular effects, we can't find it. And how is that related to the missing heritability problem? And I think they are both examples of that level of analysis problem. That our so-called non-shared, the things that, if you think about the things that make you different from your siblings, that for each one of us, that's the sum of thousands upon thousands of little non-random individual environmental events that slowly but surely over time, as nodes in a very complex system, push you and your siblings apart. But at the end of the day, looking backwards, if you say, well, what effect does having a mother who forces you into ballet lessons have on your eventual behavior? You can't find a main effect of that. It's like looking for the effect of an individual gene, because the effect of your mother's insistence is filtered through all of the other complex developmental psychological things that happen to you. So you can add them up. You can see them in gross form inside a twin study. You can see their summed effect, but you can't see their little individual effects. They're contextual. Yeah.
I study goals and motivation, so it's heartening to hear that our intentions make a difference, not just determined by genetics. And I wonder if there's an opening to maybe address this problem of the missing effects by studying people's goals, motives, and intentions. Like you have heritability for the big 5 traits. Some people have looked at motive dispositions, need for achievement, need for failure. But that's not even the main question. It would be more if you could start to look at the feedbacks between additional genetics gives you a certain preliminary motive profile, a goal profile, and then to look to see how does that, the effects of those goals then have further effects, and you're spiraling away from your genes into this world of goals and intentions and experiences. Maybe there's some way to put all this together in one model. Has anybody tried to do that?
Well, you know, I think that kind of work runs, I mean, I don't know what kind of work you do exactly, but it runs right into the no experimental control problem. That, I mean, there probably are circumstances where you can do some kind of randomization for goals and intentions, but studying it in the real world, the process of how people start out with a genotype and then develop goals and intentions which influence their behavior, it's very, very, very difficult science to do. And I mean, it's what I don't do goal direction, but broadly, I mean, that's the kind of science I do. And what I know is that the kind of findings that come out from that kind of science are changeable and contingent and hard to pin down. So it's social science. I mean, that's yet another talk I wasn't giving here, is how genomics has turned out to be social science. You know, we thought genomics was going to be the white knight that was going to kind of ride in and save us from all that difficult, not very successful science we're all trying to do all the time. But the opposite happened, that instead of genomics turning us into actual natural science, we turned genomics into social science. They're stuck with us now. Yes, Ken?
Yeah, you know, in addition to the level of analysis, explanation with missing heritability and missing environment, there seems to be another piece with the missing environment, perhaps none, but that is we don't have an environment. So that when we decide how much of the heritability include here whatever ionizing radiation, came through the airplane versus how you would read it versus whatever Lynn told you about what this talk is like. We don't have a way of parsing up all these different environmental effects, or I don't know them. And so people just look at what they think from their theories are the likely suspects. But have you given much thought to, how do you have an environmental at different levels? Because at least in the missing heritability part, I know there's a reference of, we're looking at these 500,000 to a million variants.
Yes, I mean, we know more about genomics than we know about the environment, which is, I think, what makes people at the outset more optimistic about how the genomics are going to come out. Sometimes I design a thought experiment about genes and environment to say, suppose we lived in a world that had environmental twins, so that from time to time there were people born who, for whatever reason, had the exact same experiences at every moment of their lives, right? And then those were environmental identical twins. And then there was this other kind of environmental twin who magically had exactly 50% of the same experiences in the course of their lives, right? Well, if you had those environmental twins, then there would be, just like there's quantitative genetics, there would be quantitative environmentalism. And we could use them to estimate these nice, neat percentages of how the environment works the way we can in twin and family studies. So, the point of that story is that the things that we do know, what quantitative genetics is about is capitalizing on the things we do know about biological genetics in order to explain at least some aspects of variation. It's kind of amazing that we know that, it actually turns out to be more complicated than that identical twins are twice as similar as fraternal twins. It's not often, again, I'll speak to the social scientists out there, that those of us who fit a lot of fancy path diagrams to stuff, and sometimes it feels like you can just put those arrows anywhere you want. And it's a good feeling for me because it's the only time you ever get to do it, when you draw that arc between the genotypes of the fraternal twins and put 0.5 on it. That there's an actual parameter that can get fixed on the basis of biology. That's a rare thing in the behavioral sciences. And of course, a lot of people think it's unrealistic, but the fact that you can even come close to doing it at all is a remarkable thing. So I think a lot of it's really not the point of view I was taking here. It's sort of an older version of what I'm saying, that the older view that, wow, genetics is working out way better than the environment is, because everything is turning out to be heritable. That's the world I was intellectually raised in, I think is mostly an illusion. And it's an illusion based on the fact that genomics are just more quantifiable than the environment is. How similar was your family environment to mine? How would you even get started on that?
They'd be shocked.
Twins separated at birth, yes. Yes?
So it seems like one of the, I'm a social psychologist, so my bias would be revealed here. It seems like one of the primary things about a person's phenotype that would dictate their environment is the way that they look. And so I was surprised that you didn't use that as an example. So for example, a person could look relatively more intelligent or less intelligent. That's going to dictate how people treat them, the sorts of opportunities that they get. So the stereotypes that are applied to a person as a result of the way they look can influence their environment, presumably. And the really interesting thing about that is that monozygotic twins look exactly alike. They're identical in the way that they look. Dizygotic twins, though, don't look half as identical, right? They look different. And so that could, I was actually just wondering if you would comment on that in general. I didn't mean to go on this diatribe, but it seems like that could be driving the situations in a pretty profound way. And if you could get a measurement of how similar dizygotic twins look to each other, I think that could potentially be really interesting. Have people done that?
Not that I know of. I mean, on one level, and in fact, I thought that's where you were going with this. I mean, that could be an argument against the assumptions of the twin model, that if looking alike has a big effect on how similarly you wind up getting treated, then, well, that's something that would make identical twins more similar that doesn't have anything to do with the fact that they share 100% of their genes. And I mean, there are ways you can, people study identical twins who think they're fraternal twins and vice versa, and that turns out not to make much difference. I mean, what matters is what you really are, not what your parents thought you were. As far as, I mean, where you went with that, I mean, I do think, I mean, I don't know anything about physical appearance research, but that could, I mean, I think that would be a good example of a small difference between people that could get magnified over time by the environmental exposures that it leads to. So if one member, I mean, this works better because identical twins look so alike for fraternal twins or siblings. If one twin is a little nicer looking in whatever sense one might mean that than the other twin, and then therefore the peers in the classroom pay more attention to him or her, and that makes him more socially confident, and therefore starts hanging out with a different crowd of kids. I mean, that seems like it could be exactly the kind of almost random early engine of something, but I don't know of anybody, I'm trying to think. I mean, there are studies of birth weight differences that show things like that, that the
Measurement of the similarity in twins' faces, for example. How similar do they look? How similar are they in height? And these are, of course, non-identical twins.
and that is a determinant of how similar they wind up in behavior later. Because of course, I mean, identical twins are so similar for height in the modern world that you can barely talk meaningfully. It's a rare instance that there's even a measurable difference between identical twins in height nowadays, because it's much less likely than it used to be that one twin would get very sick or be malnourished or something like that in a way that would have an effect on height. It's an interesting idea. Yes, Winnie? Last question, okay. It's been an amazing question session, thank you.
I was wondering how you fit all this into the epigenetics, like how epigenetics fits into your ideas. Does it lead to these twins getting more dissimilar?
Yep, that's right. So Wendy's asking about epigenetics, which is yet another level of complication on top of all of this. And epigenetics, for those of you who don't know, refers to a lot of things nowadays, but it refers to biological differences that are not related to your literal DNA sequence. The original idea of epigenetics is that all the cells in your body have plus or minus the same DNA sequence, except somehow that DNA sequence produces kidney in one part of your body and ears in another, even though they have the same DNA. So something other than just your DNA sequence must influence development, and there's been fascinating research done that especially traumatic environmental events, and this is not an easy thing to study in humans, but especially traumatic events in animals can produce actual changes in DNA, which can then, above and beyond that, be transmitted to offspring, which presents huge challenges. So, I mean, people often, and I think it's become commonplace these days because the biology of epigenetics has made a lot of advances in recent years. It's become an easy thing to kind of hold out and say, well, all this complexity, it's going to turn out to be epigenetics. And, well, maybe some of it will, but sometimes I feel like appealing to epigenetics is also appealing to biology ultimately when I don't want to. I think a lot of human complexity is not biological complexity, it's psychological complexity. I don't know that epigenetics is ultimately going to be the explanation of why some kids are more popular in the 3rd grade than other kids, because I don't think we need to appeal to biology to explain that kind of thing. I'm a great believer that the explanations of complex behavior are ultimately psychological. I mean, that's probably a good place to end, because that's a good takeaway of my message. Thank you very much.